Hello everyone. So today in this class we will speak about microsporogenesis. So this is the third lecture of sexual reproduction in angiosperms, that is flowering plants. So let's begin with this topic. So microsporogenesis means what? Microsporogenesis means formation of microspores. Formation of microspores. Now, what is microspores? Microspores is nothing but it is known as pollen grains. It is known as pollen grains. Okay, it is known as pollen grains. So in the last lecture I had taught this structure of TS of mature anther. So this sac, I told this is a pollen sac, and this pollen sac we also say it as microsporangium. And microsporangium is the place where microsporogenesis occurs. So this is the site of microsporogenesis. This is known as pollen sac. So total there is 1, 2, 3, 4 pollen sacs. And these pollen sacs, you can see it consists of microspore, like it you can say first of all it consists of sporogenous tissue. Sporogenous tissue. And from this sporogenous tissue, there will be formation of microspores. So microspores will be stored in microsporangium, and this is only the site of production. So this is site of microsporogenesis, and this is also storehouse of pollen grains. You should know this much. These points are important. Okay, so let's come towards microsporogenesis. So in microsporogenesis, the first thing you should know is sporogenous tissue. Sporogenous tissue is present in pollen sac. Okay, this is present where? This is present in pollen sac. Pollen sac, we say it as microsporangium. That's what I explained here also. Okay, so these cells are diploid cells, means 2n. They are diploid. Okay, these cells are also known as M.Sc, or we can say PMC. Now what is MMC and PMC? Microspore mother cell. Mother cell, or we can say PMC means pollen mother cell. So these are the cells from which pollen grains are formed. So we say it as mother. Okay, so this diploid cell, okay, this is a diploid cell. This cell undergoes first cell division. It undergoes first cell division, that is meiosis. It undergoes meiosis. So we know that there are two parts in meiosis. So let's say about part 1. So in part 1 meiosis, what will happen? This diploid cell, which is a sporogenous cell, it will convert into two haploid cell. Two haploid cell. Okay, and this two haploid cell is referred to as, like, it is like the first time division, like you can say. This is also microspore. Okay, and these two will again undergo meiosis 2. So you will get 4 cells. How many cells you will get? Total you will get 4 cells. So these are 4 in number and these are microspores. So we say microspore tetrad. We say microspore tetrad. Okay, and these all are haploid. These all are haploid. After this, what happens is there will be each cell, each microspore will undergo a it will undergo this mitosis. Now each cell we referred as microspore. Now microspore, I told that microspore is pollen grain. Okay, so each microspore means one pollen grain which is haploid undergoes mitosis. Okay, I will draw a huge diagram. So one pollen grain undergoes mitosis, and after undergoing mitosis what will happen? There will be formation of two cells within one pollen grain. One will be known as generative cell, which will be smaller in size, and there will be one known as vegetative cell. So this cell is known as generative cell and the abound cell is known as vegetative cell. Okay, so this is still here, but our ultimate goal is that pollen, we have to form male gamete, right? So after this there will be one more cell division which will be mitosis. Okay, again there will be mitotic division, so you can write mitotic division, you can write mitosis simply, and you will get this generative cell will produce two male gametes. So I can show this way that there is vegetative cell and you have two male gametes. So these are two male gametes. This is one male gamete and this is one male gamete, and this cell which I am showing in green, that is vegetative cell. Okay, I hope this is understandable. This is vegetative cell. Okay, and these two are male gametes which are haploid. Even vegetative cell is haploid. This is about how is the formation of pollen grain and the male gametes. So first of all there will be meiosis 1 which will give you two cells, and then there will be meiosis 2 which you will get out of after undergoing meiosis 2 there will be you will get a pollen grain. Okay, so we can say that there is one division here. How many division here? There is one directly I can write here meiosis. So this is mainly meiosis, you can say meiotic division. This is the first division here. Okay, this is the first division here. Okay, and then if we move forward, then you can see that this pollen grain, okay, pollen grain, which is also known as microspore, this microspore undergoes mitosis. Okay, it undergoes mitosis. So here this is second cell division, and we that is of mitosis. You should know this. And there is again one more division to form two male gametes from generative cell. Okay, two male gametes from generative cell. See, generative cells forms two male gametes, and vegetative cell forms pollen tube. Okay, so you should know this much. What is the role of vegetative cell and what is the role of generative cell? Okay, so this is like if I want to write that how many total divisions. So total division is one meiosis and two mitosis. To form what? To form, it will give you two male gametes and plus one vegetative cell. Suppose if you want to make pollen grains. Okay, so for pollen grains, for like one meiosis, one meiotic division, one time meiotic division, one time meiotic division provides four pollen grains. I had told, even I have taught the lecture of, lecture of this cell division. First basic lecture is like already taught, so go and check that. Always if there is one cell, it divides mitotically, then we get four haploid cells. If a diploid divides, so we'll get four haploid cells in meiotic division. So we will get four pollen grains. This is important question, and from here only you will get MCQ in your CET, NEET, any kind of exam, KVPY, you should know these things. So from here only the question is framed. If you don't believe, then go and check previous year questions. This is a very important thing which I am telling you, which I have marked, highlighted. Very important. Okay, this is very, very important. You should learn it. Okay, so this is about Microsporogenesis, which I have taught here. Now I will show the picture of pollen grain. See, so if you can see the picture of pollen grain, there are two layers. Outer layer is known as, means covering. Okay, see, pollen grain is covered by two layers. So the covering of pollen grain is known as sporoderm. Sporoderm means covering. Okay, so there are two coverings. One is exine and one is intine. Now what is exine? Exine means outer layer. Okay, now what is intine? Intine means inner layer. Okay, inner layer. So this intine and this exine, they are made up of specific material. Like exine is made up of sporopollenin. Okay, sporopollenin, and this chemical named sporopollenin, this chemical is actually formed in tapetum. So you should know this. Like in tapetum there are ubish bodies, and these bodies actually come and binds to the pollen grain, and hence it forms sporopollenin type of chemical above pollen. Okay, above pollen grain, like if I want to say this is pollen grain, and on top of it there is a layer which is formed by this ubish body, that is known as sporopollenin. Okay, intine is formed by two things, that is cellulose, and one is known as pectin. See, sporopollenin is highly resistant, and hence it is non-biodegradable. Okay, this chemical is actually, like, this is, we can say Exine is extremely thick. This is very thick, and Intine is thin. And why this Exine is thick, why it is resistant, it has high resistance, because of sporopollenin, and this sporopollenin is non biodegradable substance. So you can store these pollen grains and it can act as fossils because of this chemical. You should remember this chemical which is there in Exine, Tapetum, Ubish body. These can be MCQ question in your entrance exams. You should know that it is highly resistant. You should know that it is non-biodegradable. Okay, and it can be fossilized. Because of this reason only it can be fossilized. You can use it as a, you can preserve it. Okay, because of this reason you can preserve it. You can preserve pollen, and hence fossil formation can occur. Okay, so you should know this much, right? All right, so I hope sporodom you understood. And obviously, like I had told, that this pollen grain, it is not having any flagellum, it's not having any cilia. So this pollen grain is non-motile. It is non-motile. If it is non-motile, so it needs some carrier, it needs some agent to travel from male reproductive part to female reproductive part. Okay, it needs some agent. So there will be a requirement of agent, and that agent is known as pollinator. Okay, I will write it here. The agent which will carry pollen, that is known as pollinator. Okay, pollinating agent will come, which is known as pollinator, that will carry pollen grain from male reproductive part to female reproductive part. Okay, so this is about it. And you can see here Exine is the outermost layer, Intine is the inner layer, then it has nucleus, this is known as pollen grain, it has cytoplasm granular. Now some places the enzyme is discontinuous, like Exine is absent. So the places where Exine is absent on this pollen grain, that those places are known as germ pore. Okay, see, if I draw pollen, and if I am drawing Exine, see, I have not drawn Exine at this place and at this place. So these places where there is absence of Exine, okay, what is there where there is absence of Exine, that is known as germ pore. And this germ pore is the place from where pollen tube will emerge. Pollen tube will emerge means it will come out. Okay, so the pollen tube can easily come out from this germ pore. Okay, and this is your pollen tube. Now what is the use of pollen tube? As I told that your male gamete is male gamete, pollen, male gamete and pollen both are pollen grain, both are non-motile. Okay, these both are non-motile. See, male gametes, we have two male gametes. So these two male gametes are carried by pollen tube. So pollen tube takes two male gametes to the female part. When see now when this pollen tube will form, when it will form when the pollen will land on stigma. Okay, whenever the pollen will land on stigma, then there will be a formation of pollen tube. Okay, there will be a formation of pollen tube. Okay, I'm drawing a proper diagram. See, if here pollen lands, okay, so here there will be a formation of pollen tube, and here what will be there inside here, you will be having ovule. Okay, embryo sac will be there, and there this pollen tube will go and carry itself towards the egg. It will carry two male gametes towards the egg. Okay, so pollen tube is important. It is a carrier, or you can say it is a transporter. Okay, it is a transporter of of two male gametes. Okay, so this is about it. And then you can see that pollen tube, like pollen grain, it undergoes the pollen grain, it undergoes the division which is known as mitosis. And after undergoing mitosis, you can see here there is a formation of generative cell and there is a formation of vegetative cell. Okay, and this vegetative cell and generative cell starts their work. See, generative cell has formed male gametes. You can see here two male gametes. Okay, and pollen tube, if you can see here, the pollen, this pollen tube, if you can see, this pollen tube is formed by vegetative cell. So you should know these things. Okay, and this is all about the structure and development stages of pollen grain. Next, you can see the same diagram here also. I will zoom it. See, the same diagram you can see on A. You can see that there is germ pore. Okay, then you can see cytoplasm, exine, intine, nucleus. You can see pollen, like you can see tube nucleus. Okay, tube nucleus, you can see means vegetative cell nucleus it is. Then you can see pollen tube formation here. Okay, you can see pollen tube formation. You can see tube nucleus. You can see male gametes and all. So this is entirely about microsporogenesis, which I have taught. Okay. So next we will proceed towards our next part of today's class. So we will start with female reproductive world. Okay, now we will start with female reproductive world. Okay, we will start with female reproductive part. Okay, we will start with this. So I hope you are ready, and let's begin with this. So now the female part, okay, the female part is nothing but known as, it is known as it is known as a gynoecium. Okay, what it is known as? It is known as a gynoecium. Now I'm taking one picture of, a flower, and I will show you gynoecium. I will show gynoecium, and even we will speak about it in detail. Okay, I will put it here. See. So if you can see a flower, if you can see anther and all everything. So the structure which we have to speak about now is female reproductive part. Okay, it is female reproductive part. So the female reproductive part is known as gynoecium. Okay, it is known as gynoecium. Gynoecium, if I want to say a unit of gynoecium, individual member, it is known as carpel. Okay, there is one more word for gynoecium, that is known as pistil. Okay, that is known as pistil. This is the second name for gynoecium. This is the individual member. Carpel is nothing but it is individual member. What is carpel? It is individual member. Okay. So now speaking about the parts of gynoecium. So the topmost part which you can see here, which I am coloring with pink color here. So this part is known as stigma. Okay, and the part which you can see here, which I am coloring with yellow color, that is known as a style. And the red portion which I am coloring, this red portion is known as ovary. Okay, so there are total 3 parts. Okay, the first part is known as stigma. Okay, then second part is style, and third part is ovary. The place where, if you can see in the diagram, the place where pollen grain lands, okay, that part is known as stigma. So stigma is the first part, and this is the landing area of pollen. Landing area of pollen grain. If you will see the second part, that is style. So style is elongated cylindrical like part, and style is a part, you can say a passage for pollen tube. And finally you have the third part known as ovary, and this ovary is the place of fertilization where egg is present. Okay, so ovary. Speaking about ovary is a place, like ovary is a place where you will have ovules. Okay, ovary is a place where you will have ovules. So like the typical example of like the ovary, okay, there are many types of ovary. The typical example of ovary is known as anatropous, like anatropous ovule. Okay, we will study about anatropous ovule, the typical example of this in angiosperm. So let's speak about it. See, so I spoke about this, right? So now what is the role of ovary? Okay, like it has egg, fine, it has ovules, this is also okay, but ultimately what is the role of this? Okay, see, ovary converts into fruit. If you will, like in the post fertilization, we will study that ovary is converting itself into a fruit. Okay, if I draw ovary, so ovary has ovules. Okay, what ovary is having? Ovary is having ovules. So there will be ovary, like inside this is ovary, and ovary will possess these ovules, if you can see here, okay, which I am shading with yellow. These are nothing but these are ovules. So the arrangement of ovules on the wall of ovary, on the wall of ovary, this process, this phenomenon, this is known as placentation. This you would have, like you might have studied in, in 11th standard morphology chapter. So this is the process of arrangement of ovules on ovary. It is known as placentation. Okay, now let's speak about the structure of ovule. Okay, let's speak about the structure of anatropous ovule. Let's take one diagram. Okay, I will directly take one diagram because instead of me drawing and taking time. Okay, so here is the diagram. So if you can see here, this is the diagram of anatropous ovule. Okay, I have told that this is stigma, then we have style, and we have ovary. Okay, this is what? This is known as stigma. Stigma is a landing part, or you can say landing platform. And you have style. And third part you have ovary. And in ovary you have ovule. Okay, you have ovule. If suppose there is a presence of one ovule, okay, if suppose there is a presence of one ovule, then we say uni-ovulate. What we will say? We will say uni-ovulate. If one ovule is present, then we will say uni-ovulate. And suppose if many ovules are present, okay, if like there are many ovules, if there are many ovule ovules, then we will say multi-ovulate. Okay, we can say multi-ovulate. Okay, so you should know about these two terms. Even there are more two terms you can say. Suppose if the carpels, now what is carpel? It is a unit of this I had taught. See, I have written what is carpel. Carpel is nothing but it is unit of female reproductive part, that is gynoecium's unit. So the carpel, if suppose the carpels are fused in condition, like they are fused in a flower, and it can also be freely present. If they are fused, then this condition is known as syncarpous condition. What it is known as? It is known as syncarpous condition. And if it is free, then we say apocarpous condition. Apo means free, carpal. Apo means free and carpus means carpel. Carpel is the unit, like a unit of gynoecium. So the example for this, for syncarpus and apocarpus, for apocarpus, the example is Michelia, okay, and for syncarpus, the example is brinjal, okay. So you should know at least this much of examples, okay. So this is the basic which I have taken till now. Now let's proceed forward. So let's come towards this anatropous ovule, okay. Now if I want to say about, let me draw one diagram here. See, if I want to say that this is ovary and this is ovule, so this ovule, it is not present in air, okay, it's not present like this only. It is present by a stalk. It is having a stalk, okay. So there is a small stalk of this ovule which is known as funicle. What it is known as? Funicle, or you can say this funicle, or you can say funiculus, okay. This is a stalk. What it is? This is known as a stalk. And like there is also, like in between these, in between ovule and ovary, there is a presence of a tissue. This tissue is known as placenta. What it is known as? It is known as placenta, okay. You should know this. It is a tissue which is attached by the ovary, like on this tissue only the ovule develops, you can see on this tissue the ovule develops, okay. So this is the two terms which you should know, placenta and funicle, or you can say funiculus. So here in the diagram there is a presence of funicle and placenta. So this is basically attached, okay. If I want to draw this ovary, so this is attached to ovary here, okay. Here this is placenta, okay. I will draw placenta with different color. See, this is ovary, suppose ovary wall, and here there is placenta, okay. So this is placenta, okay, and then you have funicle, and then you have this entire structure is of ovule, okay. I hope this is clear. So speaking about it again, so now if you can see here, there are two parts. The uppermost part is the chaliza end, the topmost part, and the lower point where you can see a opening, this point is known as micropyle, okay. Micropyle is the place where you can see a opening, okay, small opening, and this is actually covered. There is no opening on chaliza end. You should know this much. Now the name of the structure is anatropous, okay. Now why this name is anatropous ovule, okay. Here the name is anatropous because this type of ovule is having micropyle and funiculus, like side by side. If you can see, the micropyle is actually near to funicle and the micropyle is present downwards. So here micropyle and funicle are adjacent, they are adjacent to each other, and plus they are, and plus this micropyle is downward. So you should know, and because of this two reasons, this is known as anatropous ovule. There are many type of ovule, but the basic, or you can say the general type, the common type of ovule which we say is anatropous ovule. Okay, then you can see that there are coverings. Okay, what are these? These are coverings, and these coverings are known as inner covering. See, you can see there is inner covering here and there is outer covering. So we say inner integument and outer integument. Okay, if you move forward, inner, like inside, then you can see that there is a thing known as embryo sac. This is embryo sac. Okay, embryo sac is the main, you can say this is the place where megasporogenesis occurs. Megasporogenesis occurs. Now what is megasporogenesis? Megasporogenesis means formation of megaspore. Okay, this is formation of megaspore. Okay, and this megaspore will actually, like, later it will form egg also and different cells also. Now if you can see here, chalazal end. Now in chalaza end you have cells which are known as antipodal cells. If you will see here, there are three cells present here at chalaza end. Okay, there are three cells present here. These three cells are known as antipodals. What we say? Antipodals. So I have written here Ca. Ca means at chalaza end there is the presence of antipodals. If you will see at micropylar end, so at micropylar end you will find synergids and egg. So to remember that there will be synergids, so we say MS. What we say? We say MS. MS means at micropylar end there will be synergids, and like in middle of these two synergids you have egg. Now what is egg? Egg is nothing but, egg is female gamete. Female gamete, and this egg is bigger, so we say mega. Mega means bigger. See, microspore, it is like, we can say that the microspore is, we say smaller, so we say micro. Mega means bigger. See, now why we say mega? Because here there is storage of nutrition, there is some stored reserved food for the upcoming embryo development. Okay, if you can see here light green, I will show it. This light green structure, this is known as nucellus. Okay, and this nucellus is nothing but nutritive in nature. Okay, what it is? It is nutritive in nature. Okay, so this is about the basic. Okay, this is a very basic part which I have taken of, anatropous ovule. One thing which I wanted to say, like I will add it here, the embryo sac, you can say it as female gametophyte. What you can say? You will say it as female gametophyte. Okay, so this point was left, rest the things are done. You can see here there is one good diagram of, this is a diagram, very great diagram, you can say, of Microsporogenesis. You can see here that it is starting from microspore site, okay, which is a diploid cell. Nowadays microsporocyte, that is a sporogenous cell, or you can say sporogenous cell as M.Sc, microspore mother cell, or you can say pollen mother cell, that is undergoing uh, meiosis 1, which is forming, like you can see, two cells. Then you have meiosis 2, which is forming tetrad, which I told, microspore, then it gets released. Okay, now the release of microspore occurs during dehiscence. Okay, then you have mitosis 1 in pollen grain, which where you get vegetative cell and generative cell. Okay, then you have mitosis 2. Now here they have written generative nucleus. Now why generative nucleus? Because this is a floating cell, because it's not having any cell, you know, cell wall. Okay, this is a plant, so we will say cell wall. It's not having cell wall, it's directly floating in the pollen grain. Okay, if you will, I will show you the image here also. See, there is the generative cell is having the its own membrane, its own cell wall, but the vegetative cell is actually floating here. If you can see, it's floating in the entire pollen, so we say it as vegetative nucleus, we don't say vegetative cell. See, you can see here also, this is actually floating here. This is the nucleus of vegetative cell, but it's actually floating. It's not having any, cell wall or something. So this is a good diagram. You can use it. You should draw this in your notebooks. This is a very good diagram. You should surely draw this. And you can also see that locule and all, everything, tapetum and everything. Okay. So this is about today's class. In the upcoming class, I will actually begin with a topic known as Megasporogenesis, and I will start with pollination also, the beginning of pollination. So this will be in the upcoming class, that is part 4. So stay tuned. Like, thank you for watching this class. I hope you find out like today's class useful, and soon I will come with the part 4. In part 4 we will take megasporogenesis, and further we will move forward like pollination and all. So like, thank you for watching this class. Keep studying, keep learning, keep working hard for your career. Thank you.